Thank you very much for your kind invitation, and I promise not to go over the allocated time that you've given me. It was very generous of you to give me more time. The slides about Prepare are available at this simple URL, norecopa.no/Utrecht. There are actually a lot more than the ones I'm going to be presented now, and if you just want these slides, they're at the same address but with today's date afterwards. You're very welcome to contact me at my email address on this slide afterwards if we don't have time to answer all your questions. A couple of words about the organization I work for. It's called Norecopa, and we are Norway's national consensus platform for replacement, reduction and refinement of animal experiments. We also aim to be a source of global 3R resources. And this is our website, which is updated many times a week. It aims to be an overview of global resources. There are at present about 8,500 web pages that attract about 300,000 hits a year. We also issue a very detailed newsletter 7 or 8 times a year describing the latest advances in laboratory animal science and welfare and alternatives. So, and I'll tell you on the last slide how you can subscribe to that. The background for Prepare is in many ways what I've called the burnt cake fallacy, and that is that we all too often hear that one can improve research merely by better reporting or by courses in experimental design, which apparently in some people's eyes seem to be more the mathematical issues like randomization, blinding, etc., rather than in addition to those, the animal and facility related issues. And as a lab animal veterinarian who's worked in animal facilities for over 30 years, I see all the things that can influence the validity, the robustness, the reproducibility and translatability of animal research that are related to issues that occur in the facility itself and that should be thought about from day one of planning. So what we are doing with Prepare is to try and encourage scientists from day one to approach the animal facility and discuss their project with them and emphasize, as I say, the importance of proper planning right the way through in collaboration with those who are going to be looking after the animals, rather than trying to just improve the quality of the reports that we write after the experiment, because that's far too late. And the analogy or comparison with research is obvious, I believe. I really like your infographic. It's the best one I've ever seen, Monique, and congratulations with that. It shows exactly where we believe Prepare should be placed in this flowchart, and I think it should really be. It deserves to be spread widely in our community. And reporting guidelines certainly have not, on their own, solved the reproducibility crisis. We've had guidelines around as long as I've been working in this environment. They are very important, as we'll hear later, but we need a lot more than good reporting. And equally, for an equally long period of time, there have been books and papers describing neglected factors when planning animal research. So we've known about these for a long time. We need just to put more emphasis on them. This is a book from 1994. So what I'm trying to do is avoid a knowledge gap between scientists on the one hand, who maybe aren't familiar with how an animal facility works, but who are very good on the more mathematical aspects of experimental design, and the people in my community, the laboratory animal staff, who are very aware of the effects of both internal and external factors relating to the animals and their effects on the quality and reproducibility of the experiments that are done on them. So we've been thinking about how others achieve reproducibility, and the aviation industry is an envious example. They achieve tremendous precision, and this precision is achieved in a very variable environment. This is an airfield up in the north of Norway, which you may not even have seen until I put a ring around it. And the secret, of course, to their fantastic safety record is, among other things, their use of checklists. If you talk to pilots on a 1 hour European flight, they'll tell you they'll use between 10 and 15 checklists, even on that routine flight, despite the fact they've flown it many times before, despite their long experience. So prepare takes the lessons we've learned over the last 30 years in planning animal experiments and running accredited animal facilities. The paper was published in 2017, but it was actually developed over many years, and we used it on local courses in laboratory animal science and didn't publish it because we thought this was basically just common sense, but realized after an EU conference in December 2016 that we needed to publish this. And the authors are laboratory animal specialists from the UK and Norway. Prepare consists of two things. The first is a 2-page checklist, which is available at present in 25 languages, including Dutch, of course. There are more languages coming, and let me know if you have any others that you would like to see on this list. It contains 15 main topics and a number of subtopics. There are tick boxes next to them, and that's maybe one of the things I slightly regret about the checklist, because we don't want this to be looked upon as yet another piece of bureaucratic work that scientists have to do, another hurdle to jump over before they start their experiments. Prepare is meant to be a friendly aid memoir to remind people of all the things that can affect the quality of their animal experiments. Some will be more important than others from experiment to experiment, but they're there. So we hope that these checklists will be used actively by scientists from day one of planning. There is a lot of emphasis on the 3 R's in the checklist. As I said, it covers 15 topics. I've highlighted here in pink the ones that are not typically highlighted in reporting guidelines. So hopefully you can see a bit more here about the importance of 2 complementary sets of guidelines, planning guidelines and reporting guidelines. The 15 topics are divided into 3 main groups. The first is about formulation of the study, the need for literature searches, assessment of legal issues, ethical issues, and experimental design. And then, as I said, a very important part on the dialogue between scientists and the animal facility. Is it practical to do this experiment in the facility? Do they have the infrastructure, the competence, the education? And what about the health risks? Right the way through to waste disposal and contamination. Can they be tackled at the facility? And then, obviously, an important group on the methods themselves and quality assurance there. And as we've already heard about systematic reviews, I can mention that the first part of Prepare may indeed lead to replacement, in the sense that we don't actually go ahead and do the animal study. We may be able to synthesize new evidence from the literature instead of actually progressing towards an in vivo study. In addition to the checklist, which is really just the tip of an iceberg, then Prepare consists of a very detailed website, the address at the top of the page here, and it contains links to quality guidelines and scientific papers from all over the world on the issues on the checklist, like, for example, recommendations on blood sampling volumes or injection volumes, housing and husbandry, anesthesia and analgesia, humane endpoints, experimental design. And this part of Prepare is being updated all the time as new guidelines are produced and new scientific papers are published, which are relevant to this work. So this part of it is changing all the time. And we've just added a new section for fish researchers as well, which we're in the process of populating, because there are obviously very special issues when you're working with aquatic organisms, and we felt they deserved a separate section on this website. There are a number of other things which I don't have time to go into today, which are mentioned in Prepare. One is a suggestion on how you can build a master plan to make sure that things don't get forgotten during the course of the research or during the course of the year in the facility, things that aren't done every day of the week, like calibration of instruments, but which are really important. We also have some suggestions for an agreement between the animal facility and the research group, again, to make sure nothing's forgotten, to avoid that tragic situation where a scientist comes back to a facility maybe a year and a half after the experiment, asking for data which weren't collected, which he or she thought had been recorded. And also the need for a contingency plan based on a risk assessment of the study and the facility. So there are standard operating procedures on what you do if, for example, members of staff or scientists are injured during the experiment. We also focus on a culture of care. In fact, Norecopa runs the website of an international culture of care network, which is all about improving animal welfare, scientific quality, care of staff, and transparency for all stakeholders. And we have a lot of resources on the site from the network for those who want to learn how to do more at improving the quality of care at their facility. And I thought I'd mention, having commented upon your infographic, we have a few icons ourselves, and among those is one for care, because this is a really important aspect, in my opinion, of planning animal research. So our hopes for PREPARE, as I say, are uptake on a voluntary basis. Rather than interest from journals, we're more interested in interest from people at the other end of the spectrum. That's funders, animal care and use committees, regulators, institutions, animal facilities, of course, the scientists themselves, and pre-registration, which has already been mentioned, is an obvious use for PREPARE. And we have a list of those who've endorsed PREPARE already on our website. So hopefully you won't look at PREPARE as just another set of guidelines, but that a scientist will be able to say, well, we managed to submit our manuscripts successfully because we were prepared for the questions and comments we got from the reviewers and the journals. So it's a win-win-win-win, better science, better welfare, advancement of the 3 Rs, and a safer working environment. And we made a 3-minute film explaining how all this ties together using the aviation industry as an analogy. Finally, I told you I'll tell you how you could subscribe to our newsletters. At the bottom of every page on the website, there is a white box where you can add your email address. And to remind you that the slides are available at this address. Thank you very much for listening.